Good morning.
Good morning. How is everyone?
Tired.
Yeah. All right.
Yeah. No treadmill today. I had a freak accident a week ago.
Ah.
I was walking my two dogs, and they spotted a stray puppy,
Oh.
and they ran towards it and pulled me over.
Uh-oh.
I smashed both knees.
Oh, wow.
Okay.
Sorry.
Almost recovered. A couple more days, but the first day I was on a crane.
Wow.
The cane was early.
Yeah.
Because I couldn't walk.
Yeah.
Oh well.
Yeah. So, yeah. So that's too bad. Yeah. Hopefully you get better soon.
Well, it didn't affect the brain.
Yeah. So Sushmanth has been doing some pretty good work this week. And Morgan, how are you?
Good, thank you.
Yeah, good. Great. So Sushmanth, did you want to present on your stuff?
Yeah,
all right.
I'll start with my talk.
Yeah.
Actually, I haven't done alone this all work. Actually, another guy is there, like he want to contribute to DevoLearn. He was a web guy actually, and he's not. He's so interested in open source and this all. So we combinedly did this whole work, and the idea was also his. Like before when I was started contributing to DevoLearn, he also went through DevoLearn repo and he didn't understand well. So he somehow gave up of understanding it and moved to another org. Actually, this thing happened. And this is his idea to make the whole website kind of thing, to understand the basic layout, how it works and also.
Oh, yeah. Who was this? This was, I can't remember. Is this the person who came to the meetings before?
Should I go on? Hello? Hello?
Yeah, yeah.
Yeah, this is like first page actually, explaining the tools of DevoLearn, nucleus segmenter and lineage population model. And this is a page for Hugging Face which of DevoWorm. I have added all links related to it. I have mentioned like click here, some kind of thing. And this is DevoGraph thing. Here we can go through DevoGraph documentation. Okay? These are like work have been done from 2020 to 22. Actually, DevoLearn I thought of it started in 2020 and I created this thing from 2020 to 2022. And this is like last page introduction about mentor and students who have participated in this piece of all this work. So that's, and I have sent this whole thing to my dev actually to review. He told like, yeah, that's the main problem. Like so many people are not understanding about DevoLearn or DevoGraph when they came up to see repos. So he gave me some kind of good idea to create another blog option also for DevoLearn. And in the blog option, see, like Mayuk and Minor really did a pretty good work on this thing. See, everything is like clearly documented in their GSOC time of thing. Community bonding. These all links are there for their what we call their blog pages. These are links of their blog pages, coding period week 1, week 2, week 3, week.
Yeah.
Everything is documented, but many people don't know like these things are there. So yeah, he's the Jyoti Swarup. Jyoti, can you introduce yourself?
Yeah. I know like a couple years ago they suggested that people give a weekly blog post for tracking their progress. So yeah, that's a good thing to have. Like every week you'll update it and submit it so that we can understand what you've done for the week.
Yeah. He has helped me in this whole website thing. He's from Hyderabad. He's my friend.
Oh,
we
hello.
kindly both built together this whole website. Jyoti, go on if you want to introduce yourself.
Yeah. Hi sir, I'm Jyoti Swarup. I'm an undergrad currently studying at JNTH College of Engineering, Hyderabad. Yeah, that's it from me.
Oh, great. Well, welcome. Yeah, and thank you for your work and your contribution.
And this is like, I was thinking to send these all blog pages into this website and connect a blog page for DevoLearn. Exactly. Not only Mayuk, MineckDeb also have these blog pages like their GSoC page. I will show you too.
Okay.
Okay. This is MineckDeb. See, GSoC period, week 8, week 9. Many of the people who come towards like contribute to DevoLearn, they don't know, they don't have idea like what is DevoLearn. But in these blog pages, everything is clearly explained. We have lack of documentation for DevoGraph. I will take care of it. Like I will try to add some tutorials kind of thing to understand it.
Right.
But these pages, if we integrate into this main website, it will be like a very useful tool. We can, like if anyone comes, we can directly give this web page and tell them to explore the code or tools or even like how they are built. Because Mayuk's work is like pretty awesome. He mentioned everything, how he trained the model. I mean, these are the better resources than ever I have seen. Like if I know these things before, it would really make my journey easy. I'm telling that. See, everything is like he mentioned, how he trained. He even write a blog page of like community bonding period also. So we are thinking to integrate this whole thing. That's what we are thinking. Is it okay? In DevoLearn repo only, we are creating a repo called devolearn.github.io.
Yeah.
We'll host our website there. It runs some static pages. And these all tutorials will be on like /blog. Another option will be there. And we'll make it available on the website. If someone clicks there, they can see like heading for DevoLearn done by MyDev, DevoLearn done by MineckDeb, and Gian work, etc.
Yeah, that would be great.
What is there in our mind.
Yeah, hosted in one centralized place, so we make sure it's there for
everyone.
Right.
And you don't worry about this, how to maintain this whole thing. Me and Jyoti Swarup will handle this website. We'll try to maintain as good as it possible.
Okay.
If anyone just want to write about blog, just create a PR, write a README file, like what they want, right? If they keep a pull request, if it's good enough, we'll try to add changes. If it's not good enough, we'll not merge it in the main branch. That's what we are thinking actually.
All right. Well, that sounds good. Yeah. If we could, if you want to maintain that in terms of checking, because I know Mayuk is sort of the maintainer for DevoLearn, so it's always good to have someone there who's,
Okay. And I have shown Mayuk. He was like, he told me like the website looked very fancy. Try to make it as minimal as possible, but I just want to make more interactive when someone works it.
Yeah.
I hope it is good. If you need any changes, please let me know how it's like.
Oh no, I think it's good. I think it's good.
Okay.
Yeah, I think that's a good place.
Before this work, I have talked with Ujjwal Singh actually. He suggested me the template and all kind of stuff.
Okay.
And he helped. He told to write something. And another thing is like, I have changed the format. Before submitting proposal, I have sent my proposal to Mayuk and Minak and Ujjwal also.
Okay.
They reviewed it. They told like content is good enough, and they suggested some format changes. So I changed them, and I was ready to submit. Okay, I'll go on. Like I reviewed with 3 other members, GSoCs who done work for DevoLearn. They told everything is good. Still, Minak need to give some kind of review. After his review is completed, I will submit by today night.
Okay, yeah, that sounds great. Yeah.
Sushmanth I found a book I found 2 books by that title. Is this the correct one?
Yeah. Actually, this book I was reading for my exam. Tomorrow I have an exam related to ML. This is the same book.
It is the same.
I need to start it now. Okay. There is a midterm examination is going on. This is the same book. Thank you.
Yeah.
That's are my updates. And for next week, I was thinking to work on this Hugging Face. See, wait a minute, just more. Yeah, see, our models are there here.
Okay, all right.
Here, this model is there, but we haven't had any API for this thing. I was thinking to build a blog first. After that, I will go and work on this API thing. Interactive API will be created here. Like spaces, interacting API will also be created. And everything will be hosted. That's it. Okay. I'll just look after. That's all my updates. I will let you know if any more changes are needed. Or just I need the help of creating a repo, DevoLearn.github.io. The same name organization has, exact name should be there, with .github.io.
Yeah.
That would be help. We will start pushing the code into for that as a website. So,
Yeah.
and give me, if possible, give me some access. We need to create the backend and static pages of that for that website. I mean, for that repo, please give me access.
Oh yeah, I will. Yeah.
Okay.
Do I need to give Jyoti access as well?
Yeah. For Jyoti, for that repo itself. For me and Jyoti, please give that.
All right.
That's all of my updates. I will let you know if anything else.
All right. That's great. Thank you. Thank you for the update. Yeah, and glad to see that we're working on this. So that was something. The GitHub.io is something we've been building for a couple of years now. We in 2020 when we did DevoLearn, we kind of had all the models. It was kind of a place where we collected a lot of the models and then put them in that place. That was actually before we really started the DevoLearn trajectory. So it was pretty out of date. But I like the idea of having, updating it to where we are with DevoLearn and DevoGraph, and then having like these tutorials available so people know kind of they have, a lot of software has like tips and tricks, or they have FAQs or something. This, I think, is a good way to do that. So, yeah, and then we can, continue. I would recommend that for people doing GSOC that we do weekly blog posts to keep updated. INCF used to require that for GSOC, but last year I don't think they did, so we didn't do it. But I think that's a great idea. So keeps everything, keeps everything you do in context. It's like we can see your progress as you move along. So that's great. And then I'll give you permissions to that repo because it's different from the DevoLearn repo in a different organization. So great. Thank you. Go ahead.
Yeah, nothing. I just unmuted.
Okay. So Susan, how are you?
I'm good. I've been told that COMSOL's useless and that I should do my model now in MATLAB. We'll see.
Yeah. Why is COMSOL useless?
It won't give me the precise matrices. I need to somehow see the matrices to see why my model is failing. I change a parameter and it fails. So I'm pretty much assuming that parameter causes it to fail, but I need to know exactly. Or it's useless, apparently. So I get to try this in MATLAB. I'm still holding out for more Morpho from Timothy, who I met in the APS Physics. He gave a lecture in my workshop on imaging, and he runs something called Morpho, and it's from Tufts University. Yeah. So, and they've been doing some of that hexagonal, they take spheres and pack them, and they end up with hexagons on a flat surface, and then if they're on a curved surface, then there's gaps in it, and then they've got something that looks like a oblong that looks like a fly embryo that they're working with. It's quite interesting. Anyway, they use Morpho, and I'm hoping that might be somewhere between COMSOL and MATLAB somehow. I can get a model in MATLAB, and I can port it into console. I don't know. Well, I'm working with different things now, platforms. Yeah. Hurrah.
Yeah, the challenges of modeling, I guess.
Yes, except that I do have my model. I'm fairly sure that the one that I put together is the correct one to try, like the correct physical configuration to try tensegrity out in cells. I'm pretty sure of it.
All right.
Yeah, including the fact that the elasticity in the actin filaments is quite stiff. So you get a J curve with the tensegrity when the actin or elastic filaments or the strings in your model are quite stiff. It seems to work that way. I have a paper about someone who did that with the triangular tensegrity, and that's the way it worked. And I think I presented that.
Yeah, you did, I think.
Yeah, so there's that. And then I added the actin ring to my model, and then I added a tensegrity link between my parts, and I think I've got something that behaves more or less cell-like. I'm also in the APS physics. They were going about hydrolonic acid and brush borders and how integrin just attaches at certain places and not all over the place in a cell because you've got these brush borders. And I guess they're also maybe along the sides. So the attachments really are just at the corners of your cell. Whatever corners the cell comes up with, they change.
What are brush borders?
Yeah, I actually captured the guy's lecture. I went, they have them, the lectures after the conference. They have some of the main ones, and this fellow's lecture was there, and so I captured a couple of images. He didn't want it recorded, so I was careful.
Yeah.
But I could show you. I mean, I could show you what he means, but it's nice. It's basically there's just these little brushes that grow of hydrolonic acid. And so the attachments only occur at intervals. They're not all the way across the base membrane. The attachment to the ECM is only in some places because of the brush borders growing.
Oh, wow.
Yeah. So I think my model should be good.
Yeah.
It's certainly worth a try.
Yeah.
If I can find out what platform to put it on.
Right. That's so Morgan mentioned Phoenix, which is a method. I guess that's a multiphysics model, or no, for finite element analysis.
Okay, yeah.
But,
well, I have. Yeah, I'm trying to find something that will give me the matrices produced by the finite element analysis, and when it fails, I want them to say why. That all I'm getting out of console is it's an empty matrice. Morpho, actually, Timothy Alberni, I think it is, from Tufts, said he would try it. He said he thought my model would work. Yeah, I want to download Morpho and use it now. I was like tired of some of this other stuff. But anyway, I get to try MATLAB this week.
oh yeah.
I actually, there's a book written by Gan. The guy's last name is Gan, and he has pre-code. He has code that I can use. Actually, I've used it and made my model in MATLAB, but it's the twisted model, a standalone twisted tensegrity.
Right.
So I just have to straighten it out and put some blips on the side and add a layer. So I get to try that this week. If I can do that this week, it'll be good. Then I can show the professors that I did something. And in the meantime, I'm going to try to download Morpho.
Susan, it's been many years, but I have a vague recollection of the property of hyaluronic acid, which may or may not be relevant. Property of hyaluronic acid.
Oh.
Yeah, if you can, apparently, as I recall, if you twist the molecule, it can change between hydrophobic and hydrophilic.
Oh, really?
I'm not sure. I can't find the literature right now. Okay.
Oh, well, I have pictures of brush borders from.
It's a vague memory, but it's a fascinating property.
Yeah, that would be interesting.
Okay. I used to correspond with a fellow who studied it, but I can't find anything. I can't even find his name.
Oh, well, that would maybe line up with things if your cells get twisted because they're trying to change places, and all of a sudden they become hydrophilic, and they go, whoa. I don't know.
It's a wild property. But, I think it was hyaluronic acid.
Okay.
Okay. But it's going to take some hunting to find the papers.
Okay.
Yeah.
Do
So
you want me to look for that?
No, it's up to you.
Let's
If
see.
you find them, let me know.
Okay. That's fine. Just carry on, and I'll look for this.
okay.
Okay. Bradley, just so you know, I have a meeting with Tom Porteous at 10.
Okay. All right.
Okay.
Yeah.
Probably disappearing.
All right. So yeah, Morgan and I actually had a nice talk this week about multiphysics models and finite element analysis and kind of the things that he found. He's finding a lot of the stuff we're talking about interesting, and he was inquiring about that. So, and I recalled that we talked about finite element models like very early on in DevoWorm. If Dick remembers, like 2014, 2015, we talked about that. I was even back with Steve McGrew when we were thinking of using that as a method. And never really happened, but it was that sort of that type of a model. So
yeah, I think the most fascinating part of that is Wayne Broadland's stuff doing the inverse finite element model, where he could use finite elements to measure the forces between cells and embryos.
Yeah.
Okay, and he's retired and he's gone off and he's not doing any more science. So it would require resurrecting that stuff from the papers.
Yeah.
That's interesting. That's kind of how it's used in high density EEG, at least it's mostly used for inverse problems. Yeah.
What is EDG?
Electroencephalography.
Oh, okay.
So
that's an inverse method, huh?
Yeah.
Interesting. Okay.
That's what they want to do in with what I'm working on is inverse. They want to do the inverse method with it. So with the tensegrity, you've got something that's in compression. It has one value, and something that's in tension. It has another value, And so you can more easily do an inverse method with two unknowns. Anyway.
Yeah. So we were doing skull conductivity estimation by injecting current into the head. That's what I was doing, or that's what scientists were doing at electrical geodesics for these head models. But I'd be, yeah, it does sound like COMSOL's not working right if you're getting empty matrices.
Well, that's only if my model's not working. I can get it to work, but trying to figure out why it's not working. Like I can take it to the edge of where it works, and I actually have one set of variables that sometimes gives me a result and sometimes gives me an empty matrix, just depending on maybe which hand I use to push the button. Like it's just like it runs out of iterations, and I looked at the iterations and they're going to hell, and it's converging, and then bing, it goes way up here. And then maybe it starts down, or maybe it starts to bounce around. Anyway. Yeah, I saved the iterations because it was so unusual. Like this. You think it was converging, and then all of a sudden it doesn't.
Oh, yeah.
But I'm going, well, it's a tensegrity, so of course they're different. Fractal, chaotic, / in that range of things. So
Right.
yeah. Well, I guess I should expect that, but wild. Anyways, if you want to see my screen, I can briefly show you what the guy was working with.
Yeah, it'd be fine. Be good.
Just briefly, because yeah. Okay, thank you then. Sorry, just a minute. I'll get there. Okay, share screen. We'll hope that works. There's the share screen. Window. Okay, there it is. Can you see that?
Yes.
So this is this brush that grows. It's a glycocalyx, and it alters adhesion, and it's in here, and you can only adhere to the ECM in some places because of this brush.
Right.
So it without this brush border, the glycocalyx, the cell is flat.
Okay.
And with it, then it has more of a shape. This sort of thing happens.
Right.
Yeah. So
does it allow it to separate from the surface selectively, or is it just?
Yeah, I didn't get that. He had trouble getting his slides up and going, so this is all we have.
Okay.
It adheres, and then it attaches. And I've got my model does this.
Okay.
Where it's attached just at the edges, and I think it's a legitimate cell model because this happens in cells. So
yeah.
I mean, I can say, okay, this goes on, so that's why my model is okay because it's just attached at the edges.
Yeah.
It might attach in the middle a little bit too, but just as a beginning model, it should be fine.
Yeah. It's differential adhesion and accounting for that.
Yeah. So I just this is what he presented.
Well, that's great. That's great. Yeah.
Anyway, okay. Have I stopped sharing?
Yeah. Yeah.
Okay.
All right. I put Wayne Brodlin's relevant papers in the chat.
Yeah, that looks interesting.
Okay.
Several papers. There's a review on finite element methods for developmental biology. That's this one from '94.
Yeah, I should read that one.
Yeah.
Sure.
And and then there are a couple others in here that are interesting. So yeah, it'd be great. Read through those and revisit finite element modeling. It's kind of a beast to get going, and yeah. But
yeah, it comes from. Let's see if I find it. I did a paper. A guy named Stein.
Anyway, Morgan, do you know anything about downloading and running Morpho?
So is that a MATLAB toolbox?
No.
Oh, okay. I haven't checked it out yet. Sorry, I'm in the car.
Well, I think it's Python.
Oh, well, even better. I mean, I have a MATLAB license, but I try not to use it.
Oh, okay.
Well, it's just it's really about sharing models, you know. So I just don't like to assume everybody's got MATLAB, but
no, it's expensive.
yeah. It's so super expensive for something that's basically a scripting language around, BLAS and LAPACK, but I'd be really happy to check it out if you've got a link to it. But it sounds like Tufts is
it's just
the download. Yeah. But yeah, I'd be happy to check that out. Maybe talk with you, work with you if that would be helpful.
Well, thank you. With COMSOL, I was basically out on my own.
Yeah, I mean they're pretty complicated packages to use. You've got a benefit in head modeling that there are some packages for it specifically in my field. That build on top of packages like Gmesh and others that are commonly used. It's sort of like an alternative to Phoenix. But very interested, and the Tensgressity in particular. I'd really like to see how to try and fit that into a model, and again, I'm really interested in trying to transfer this knowledge to more like cellular work.
Yeah, well, it's just a matter of building a tensegrity, and it's just tension elements, which are basically strings, and compression elements, which are rods, and you just make the rods fall apart in tension. Like they just do compression, and the tension elements just do tension. Like they're a rope. You can't push a rope. So.
Yeah. I'm just mostly doing bioelectricity, and whereas I know that these models are more commonly used in mechanical engineering. So I've got a bit of learning to do, but really interested.
Oh, okay. Yeah. I'm interested in mitochondria and that electrical gradient that this DevoWorm did one day. Anyway, so I want to know how the electrical gradient interferes with light.
Okay.
Okay. Susan, we had one weird result which we could not reproduce in the lab. If you made a coil, so it generated an electric field, and then hatched axolotl embryos in it, once they were all aligned. Okay.
Oh,
And we couldn't get the results to happen twice.
no. Well, it's axolotl embryos. They do what they do, and they sometimes do very strange things.
Very perverse embryos.
Yeah, yes.
I think another time we tried to put zebrafish embryos in an electric field, and it looked like cells were coming out of the embryos.
Oh, well, they probably were. I understand they're loosely packed in some
I
parts
don't know, but
of development. Yeah, their tail, maybe their tail part was.
yeah, that's the full extent of my direct experience with electrical fields and embryos.
Oh, okay. Yeah, my background is electrical engineering.
Okay, I put if you got people who can't afford MATLAB, but Octave is apparently similar.
Yeah.
Yeah, Octave. Yeah.
Yeah.
As a grad student from the university, I actually get mine for free
Right.
or semi-free.
Yeah.
Like $100 a year or something. It's like
Okay,
very.
that's less than one module.
Yeah. I've tried to buy it when I was teaching it. They think I'm going to teach it, so like I wasn't teaching it. No.
Well, thanks, Susan, for that update on your work on the 10-site ready work. Yeah, we can revisit some of this stuff. If you have some ideas for modeling, want to try some things, let us know. Yeah.
Just got to find the right finite element analysis platform so that I can see the matrices and see exactly why the model's failing.
Yeah.
I can take a guess. I'm pretty sure I know why it fails, but no, I have to know exactly why it fails. Yeah.
Okay, I added one paper that was done. Stein was a medical student at the time. We did this paper on epithelia as bubble rafts, and we only examined real bubble rafts. In other words, we made bubbles between two glass plates, okay, and then measured their curvatures. And that's the paper from which Wayne developed his technique. He generalized it. We did a very simple case, and he generalized it to all epithelia.
Oh, okay.
Yeah.
Is that more or less what Tufts is studying too? Is like the spheres compressed spheres. It's on their website, and they find these that it turns into hexagons, of course.
Yeah.
Yeah.
Well, here we're looking at the curvature of the bubbles.
Okay.
And the curvature is a measure of the pressure inside the bubbles. And for a bubble raft, if you have two bubbles up against each other, the net result should be zero, which is what we got.
Yeah.
Okay.
Okay. I'll look at that. That's because I got asked why hexagons. I'm going.
Oh, why hexagons? Okay.
Because Ingber used it, and like I could have listed a whole pile of other people who were using epithelial as hexagons.
There might be stuff on proving that if you have bubbles, a flat layer of bubbles, that they have to form hexagons.
Yeah, well, that's what Tufts was working on. They have a paper on it.
Yeah. I mean, we use a flat layer of bubbles for this work.
Yeah, they did that. So, and they modeled it. So I'm going, everybody's working on the same thing. But no, it's there. I'll get you the paper. Okay, well, as far as
he was a very bright student, and it's one of my great disappointments because he became a psychiatrist.
Oh, okay. I hope he had a good bedside manner.
So I'm going to share my screen and talk about some items. of interest. This is an interesting piece that just came out in APS News. So we're talking about APS again. This one is an opinion piece by William Bialik, who's a big name in biological physics. And so he's reflecting on biological physics coming of age. So this is brand new. And he's basically recounting, how biological physics started and how it's now coming of age. So once an awkward...
How does he say it started?
What was that?
How does he say it started?
I don't know. We'll see in a minute here.
Oh.
Yeah. Once an awkward confrontation between disciplines, biological physics is having its moment and showing that life is not just a mess. So he kind of goes back to the 18th century for some of the origins here. So physics and biology were not always separate disciplines. In the 18th century, controversy about animal electricity were foundational for the understanding of electricity more generally. So this is Galvani and his experiments with muscle and electricity. And even Darwin, I think, did some experiments with electricity and muscles. So that was something that people were really interested in at the time. And it's, but that's something that before physics and biology really became came into their own as disciplines, I guess. In the 19th century, explorations of vision and hearing intermingled with the emerging understanding of optics and acoustics. So that's another kind of point of intersection there. In the 20th century, the chasm between physics and biology grew wider, but there were spectacular bridges. Most famously, the work that launched our modern view of life, structures of DNA and proteins, theories of base pairing and genetic code were done primarily in physics departments. So, and still today, even, a lot of the work on some of the tools for genomics are done, in like maybe a biochemistry department or somewhere else. You have computer science departments even. So it's kind of interesting how these things migrate to different fields and, provide these opportunities for interaction between fields or between departments. So the revolutionary successes of reconnecting physics with biology in the mid-20th century completely changed how we think about life and even changed how biologists' work is organized. So I guess what he means by that is like the genomics, or at least the revolution of molecular biology in some of the things that people were doing in physics departments with X-ray diffusion and things like that. And so what emerged first was called molecular biology. And over the course of a generation, the ideas and methods of molecular biology, grounded in physics, touched almost every part of the biological sciences, even physics. So then, he talks a little bit about his awareness of this as a student in the late 70s. For me, the things biologists talked about were interesting, but the way they talked about them was unsatisfying. Physics was the other way. The style of thinking was attractive, and the theories elegant and powerful, but I never had an original idea about problems in the field's traditional core. So, that's true, I think, in a lot of fields where people have this sort of urge to be interdisciplinary because of maybe the problems they're interested in. They don't necessarily fit into the core of the field, but also, sometimes a field like, biology, they don't really talk, like I'm thinking of computational biology for me. In a traditional biology department, they don't talk about the problems in a way that's satisfying because you're always thinking of computational aspects of the problem. Whereas, in computer science, for example, you don't usually, the idea of, biological computation or, biology or computation for biology isn't at the center of the field. It's kind of like this thing on the edge of the field. And so you're going through all the stuff that you find extraneous to get to the thing you're interested in. So yeah, I have a similar experience to this, but with computer science. And so it was clear that physicists were doing all sorts of interesting things connected to the living world, but these efforts didn't cohere into a community, and certainly not into a recognizable branch of physics. We went to meetings where mostly we would find biologists working on the same systems, but not physicists asking the same kinds of questions. If we asked our biology professors, they would argue that a physicist's search for simplicity and universality was an obvious conflict with the complexity and diversity of life. If we asked our physics professors, they might talk about a colleague who had become a biologist, and perhaps warn that experiments in biology are messy. Discussion with both groups could easily be less polite than I render them here, but they did agree that it was a waste of time to try to do theoretical physics in the complex context of living systems. So this is kind of like when before really biological physics was a quote unquote thing. And so, community kind of was people kind of moving between fields or, whatever. And then I guess now it's becoming sort of a newfound relevance, I guess. So 40 years later, which was, I guess, now, or in the recent past, the intellectual landscape has changed radically. At the APS March meeting, we see physicists attacking an enormous variety of problems drawn from the living world. They wrestle openly with the complexity of biological systems while searching for general principles. More and more young people are entering the field, and our community is no longer trapped at the interface between disciplines. Biological physics has emerged as a branch of physics, and so now it's kind of this a little bit more coherent thing. So let's see. You had a comment in the chat. So
I did heart electrode impedance when I was in third year engineering. You want to know what year that was?
Was it before or after 1990?
It was in 1970s, late 70s.
Okay.
Mm-hmm. Yeah. So there was some biological physics going on.
Yeah. So that Dick puts in the chat, given a spherical cow, the physicist's view of life. So that's a problem we've talked about a lot in the meetings. The spherical cow, which of course is where there's a joke about physicists modeling something, in biology, and they say assume a sphere in a vacuum, and that's always like the first step to modeling.
Okay. Bashir Ackman and I have this submitted.
Oh, the paper, yeah, in the chat. Okay. Yeah, this is it a Janus-faced world after all? Physics is not reductionist. Okay. And this is the new paper on Janus-faced.
Yeah.
Yeah.
We'll see how the reviewers say it.
Oh, okay. So it's not there yet in terms of.
No, it's not reviewed yet.
Okay. Yeah. Yeah. So I mean, yeah, this is a nice overview here of so we have these genuinely new physics that we can look at. We have different biological processes we can view anew as physics problems. So the message of biological physics is that beautiful phenomena of life connect to deep physical principles. So things like bacterial swimming and sensing, you can view those as being dominated by low Reynolds number mechanics and diffusion, and that gives you a new insight into these behaviors. We've talked a lot about that in the meetings here where, we're talking about behaviors and the physics of cells, the physics of, single cell organisms. Vision and photosynthesis illustrate how quantum mechanics can produce a broad absorption band rather than narrow spectral lines. So in photosynthesis, people are interested in quantum effects, and in vision, there's a lot of like physics and psychophysics and regular physics. And then DNA exemplifies the random polymer that appears in all statistical mechanics courses. So a lot of like molecular diffusion and a lot of things that involve, biochemistry have this sort of statistical mechanics flavor. And we've talked about statistical mechanics a bit more broadly than that even in this group. So that's something that is now getting some more attention. So that's an interesting article on that. Then, Susan, I guess she sent out an email this week talking about tissue flows and things like that in the embryo, and I think they were covering that at APS Physics as well. So there's a press release here from Columbia Engineering, it's their mechanical engineering department, and it's a new view on how tissue flows in the embryo. So this is Columbia engineers developed novel technique to understand the forces that control cell shapes and tissue flows in Drosophila embryos. And so this is an example of so in the Drosophila embryo, you get this huge syncytium full of, it's a single layer and it has a bunch of nuclei in it. It's multinucleated. And then that's something they call a syncytia. And then that syncytia cellularizes and becomes all these cells. And so then these cells move around and they flow in the embryo. So there are forces going on in the embryo after cellularization that moves the cells, and it starts to control shape and the position of these cells, and you end up that goes to later stages of embryogenesis where you get structures and other things in the embryo. So if we had another message here, Morgan said interesting. Susan cited the person Timothy Atherton. Okay. So this is on a new paper that was published. So basically, they want to know whether thought we might be able to learn something about whether the embryonic tissues are solid or fluid just by looking at the shapes of cells in the tissue. So I guess this means that they want to know if it behaves like a solid or a fluid. So we decided to try this in the fly. We were really excited about our results, which could reveal fundamental mechanisms underlying human development and point to where things can go wrong, causing birth defects. So this is, again, you have to use traditional engineering approaches, but you want to be able to measure these mechanical properties and see what tissues behave like solids, maintaining their shape and resisting flow, and which tissues behave like fluids, flowing easily and changing shape. So you have these flow fields in the embryo. You have cells that are sort of compliant to those flows, or cells that resist those flows. And so in this study, they focused on a very fast-moving developmental event in which the embryonic tissue rapidly changes shape to elongate the head-to-tail body axis of the fly. So these Drosophila embryos are long, they're oblong, very oblong actually for an embryo, and they have this polarity early on. And these flows go from anterior to posterior. So we have this have a little animation of these dramatic tissue flows here. These are epithelial cells. And they're kind of moving and they're elongating this head-to-tail axis, this anterior-posterior axis. So this is nice, a nice set of studies here. So
are they saying that the cells generate the force, or a force is causing them to move?
I think both. I'm not really sure how the force is generated, but I think it has something to do with like the sort of the shape of the embryo, or like there's some cells that are making making forces, and then those are transmitted across the embryo. I'm not really sure exactly what the mechanism is, but do you know, Susan?
Pardon? Sorry?
Do you know what the mechanism is for the cell flows? Like what the origin of the forces are?
Not really. There's different forces on the sides of the cells, and if you get enough force occurring, you get some of the cells, the sides will close together, and you'll get what they call a rosette. And once you have a rosette, then it'll shift. And what you do is you get a shift of cells this way to cells that way. It's, what's it called? Convergent extension.
Oh, okay. Yeah. So it's a convergent extension property, just kind of like generating it. Okay.
Yeah, but it originates from different different forces on those nodes, because in my cell model, if you hold it with just 2 nodes, you get an elongated structure. If you hold it on 4 nodes, you get a square or a rectangle. And so it's dependent on the forces on the nodes, on those actomyosin complexes that are sit right on the corners of an epithelial structure.
Okay. So this
I
is
can show you.
so this is in the tensegrity model.
Yeah. But this follows through to any structure you make.
Right.
Of a cell. It's a hexagon. And how can that change from a hexagon? Well, one way is you just have different forces holding up the actomyosin nodes on the corners. That's one way you can do it. But the cells also eliminates their sides. Like instead of a hexagon, they'll become a pentagon and then a square. Like they regulate themselves.
Right.
And they'll do it fast.
Yeah.
That you can, if they're going to move, they move.
Yeah.
I'll go get that.
All right. So yeah, we actually last week I did a thing. I do it after the end of the official meeting, and I cover some papers on the YouTube channel. So those are something I want to check out at the end of the last recording. What the papers were. We did I did a couple on curvatures and condensates, and basically it's this idea of curvatures in cells. And there we go. There's the
there's my foolish model.
Okay, yeah, that's great.
You're
holding it up.
Well, sorry, I'll just try to do this quick. Sure. I don't know how I'm doing. Anyway, if you hold it up just at two nodes, you get this structure.
Okay.
Yeah, it's more or less a rectangle, isn't it? That's
Yeah,
just held up 2 forces.
Yeah.
On opposite sides. So change of shape depends on the forces you're putting on the nodes.
Right.
And they actually had some papers on this at the conference.
Okay. Yeah.
I'm not sure why I get to do these things, but anyway.
So yeah, last week did some papers on curvatures and condensates. So people were looking at different macromolecules in a condensate, like they were using a water droplet, and they were looking at some of the organization of things in there. And then they were looking at Drosophila embryos and looking at this problem of cellularization and then these fluid flows. So this is the common problem of what happens to these things in a flow. Do you get actually I think they were also using bubbles in this condensates paper. So they were looking at bubble formation, bubble transport, and then the other paper they were looking at cellular movements in the Drosophila embryo. So this week I wanted to talk about 2 other papers, and these are basically Drosophila embryo specific. So this is the first paper, anisotropy links cell shapes to tissue flow during convergent extensions. This is what Susan was talking about with convergent extension. They don't talk about tensegrity models in this paper, but they're talking about this idea of tissue flows and cell shapes in convergent extension. So the abstract here reads, within developing embryos, tissues flow and reorganize dramatically on timescales as short as minutes. This includes epithelial tissues, which often narrow and elongate in convergent extension movements due to anisotropies and external forces or internal cell-generated forces. So this is where they're elongating according to different types of forces and different orientations And so, that's kind of like, some set of forces or force interactions will trigger this. You get this convergent extension, and then you get these cell flows that result from that force being generated.
Mm-hmm.
However, the mechanisms that allow or prevent tissue reorganization, especially in the presence of strongly anisotropic forces, remain unclear. So this is something they don't really, this is what they're going to answer here, but it's probably still remains unclear. We'll see. We study this question in the converging and extending Drosophila germ band epithelium. So there's this germ band. Actually, I have some Jamboard images here of this. So this is where you get this cell flow, you get convergent extension, and you get these flows that move cells around the embryo. I don't actually know if I have an image of this, but okay, I don't. Basically, it's where you have the Drosophila embryo, and you have the furrow moving across it. We've talked about the furrow before. And you have...
Okay, can't see it.
Oh, yeah, I'm drawing it now.
Oh, okay.
So this is the Drosophila embryo, the cellularization, and you have this furrow moving across it, and you have germ bands that form in this structure, and so you're starting to get, structure moving across the embryo. And so this is just the context for this, where you're going to see these cell flows. You see them at the edge of the embryo, but you also see them inside the embryo, and it's going to start moving things around. So this is, so we study this question, the converging and extending Drosophila germ band epithelium, which displays planar polarized myosin II and experiences anisotropic forces from neighboring tissues. We show that in contrast to isotropic tissues, cell shape alone is not sufficient to predict the onset of rapid cell rearrangement. So they first test this with cell shape, and they figure out that it's not sufficient to predict this. From theoretical consideration and vertex model simulations, so this is where they're doing some simulations, we predict that in anisotropic tissues, two experimentally accessible metrics of cell patterns, the cell shape index and a cell alignment index, are required to determine whether an anisotropic tissue is in a solid-like or fluid-like state. So they basically want to know if something is in a solid-like or fluid-like, like they mentioned in the preview article. They have to determine the physics of the state of the tissue. We show that changes in cell shape and alignment over time in the Drosophila germ band predict the onset of rapid cell rearrangements in both wild-type and snail twist mutant embryos. So the wild-type are the ones that are normally developing. Snail twist have some mutant phenotype. And so you can look at this in different ways, where there may be differences in the process. So then where our theoretical predictions are further improved when we also account for cell packing disorder. So this is where there's disorder in the cell packing. These findings suggest that convergent extension is associated with a transition to a more fluid-like tissue behavior, which may help accommodate tissue shape changes during rapid developmental events. So this goes, if you go down to the figures, they show this germ band epithelium here. This is the embryo. You have these different parts. This is the part that they're highlighting in the box. This shows the germ band epithelium down here. So that would be down in this region here. And then this shows a little square of that epithelium. So you have an internal stress from planar polarized myosin II. So these are myosins in here, and external stress from neighboring tissues. So it's really internal to the cell and from across cells. So cells are interacting collectively to generate some of these stresses, but they're also working against internal stresses generated from myosin, and that gives you this sort of...
Yeah, there's some studies on muscle bands in developing fly embryo, and this sort of looks similar to that. It'll create structure, so it's interesting.
Yeah. And then you have the cell rearrangement and cell stretching. So cell rearrangement is where you get this sort of rearrangement of sort of shape. So here you have this these two cells across, and then one cell up here, one cell down here, and then over time they rearrange so that you have these cells touching, these top and bottom cells touching each other, and the middles and the cells in the middle here in this middle band are forced outward. That's cell rearrangement. It's basically where they're pushing together and they push each other out of the way and they rearrange like that. Then there's cell stretching, where you have the cell packing that stretches in shape. So it's just stretching, but it's not rearranging in any way. And so they show here, this is axis elongation over time. You have relative anterior-posterior tissue length and cell rearrangement rate, which is this relative AP cell length change. So you have two y-axes, and I think the y, I don't know, the purple and the blue. So the purple here is cell rearrangement rate, and the green is relative AP cell length change. So they're different y-axes per color, and then this one is, those are equivalent to relative AP tissue length. And so basically the bottom line of this is that over time, the anterior-posterior cell length change decreases when we get towards 0, and then increases after, I guess this is the event that they're measuring. Before the event, you have this, it kind of goes to 0, kind of goes below 0 at the time of the measurement, and then up. And then it settles back down to 0 after, like about 30 minutes after. So this whole event takes place in this period of time. The cell rearrangement rate, however, stays at 0 until about 5 minutes after the event, and then goes up, and there's a peak around 10 minutes. And this is, of course, probably where you're getting a lot of this flow, reactivity to the flow. So you can see that there's stuff going on about 10 minutes or to 20 minutes after this measurement, and so you're getting, this is the effect of this sorting because of this flow of tissue, or this tissue flow effect. So there's another figure here where they kind of show this number of cell sides, vertex coordination number, and they do these vertex model simulations. So this is where you get different fractions of different cell types. So you get 5, 6, and 7. You get this fraction of pentagonal cells on this axis. You get the average shape index, P bar, on this axis, and you can see that they're simulating a packing of cells, and they're showing these simulations. So they have the fraction of fluid tissue states going from 0 to 1. Near 0 is blue, near 1 is red, and you can see that they go. It goes above this regression line in red, and it goes below the regression line in blue. So there's this fraction of fluid tissue states that's lower, as we go below this trend line, and higher as we go above, which is kind of interesting. If you look at this for 5-sided cells, you see this, the blue are down here and the red are up here, and then the vertex coordination, the red are up here and the blue are down here. So this is kind of interesting. I don't know what that means exactly. Let's see what they say in the legend. Cell shape and packing disorder alone are not sufficient to predict the onset of cell arrangements in the germ band. Confocal images from time-lapse movies of epithelial cell patterns in the ventrolateral region of the germ band tissue. That's what we're looking at here, or we're analyzing. Cell outlines were visualized by using the fluorescently tagged cell membrane marker GAP43M cherry. So this is actually up here where they're looking at the images, and then they're analyzing these images. So they don't really tell us much about this. In model tissues, we find a linear dependence of the critical cell shape index on the fraction of pentagonal cells, which is a metric for packing disorder. The dashed line represents a linear fit to this transition. The relationship between P bar and F5 for 8 wild-type embryos, with each point representing a time point in a single embryo. The dashed line is a prediction from vertex model results the relationship between p-bar and vertex coordination number for 8 wild-type embryos, with each point representing a time point in a single embryo. The dashed line is the prediction from this reference here. Instantaneous cell rearrangement rate per cell in the tissue is represented by the color of each point up here, with blue indicating low rearrangement rates and run to yellow indicating high rearrangement rates. So basically you have low rearrangement rates versus high rearrangement rates. And they find that if you look at the shape index that predicts these rearrangement rates for different size cells and different, so these are the pentagons here. The vertex coordination number is where you have how many vertices that when the cells are in a packing, what size vertex forms. If you have 3 cells that are coming together, it's 3. If you have 4 cells that are coming together, it's 4, and then 5 cells coming together, it's 5. And I guess the implication of that is that you get more opportunities for these forces to generate a flow. If you have 5 cells packing together, they're all kind of moving against one another. And then there are also these forces that are being transmitted across the population of cells. So that's kind of what this is. I know that's not as clear as we'd like it to be, but that's...
Well, you should be able to do a simulation of that somehow.
Yeah. I don't think they did one here. I think it's just that they're taking that data, they're analyzing it, they're using the function from some other paper, and they're saying this is the sort of what we predict. So they're not really doing any simulations there.
Images are good.
Yeah.
I like images. Then I can say, well, see, that's an actual cell group.
Yeah. So this is the vertex model, and then this is increasing strain. So you have this box or this field of cells, and then you're compressing it as we go down. So this is increasing strain. And then this is the effects of strain on model tissue. So this is this average shape index p-bar, and then against the shear modulus, which is G. So now we're looking at shear forces, and we're looking at this increasing strain as we look at these different points here. I guess these are different cells in these packings. So this is the blue packing here. This is the shear modulus for a wide range of values, and then the average shape index is low. As we get increase the strain, in this case the purple, we see that it's for a lower range of shear moduli, and then it's a higher shape index. And then for the red, which is this tightly strained or highly strained packing, you see these red points, which are a little bit less shear modulus, maybe about a half of the range from 0 to 1, but you get an even higher shape index. And so this is where, this is kind of an interesting result. Again, I don't think this is simulated. This is just demonstrating what's going on. And then you get this cell shape alignment, or Q, versus average shape index, or p-bar. And so you can make predictions for different values of these parameters. So for high p-bar, low Q, this is what you should expect. For high p-bar, high Q, this is what you should expect. And then low p-bar, low Q, this is what you should expect. I think this is looks like a lot of 3 and 4 vertex points in this matrix. In this matrix, you have a number of 5 vertices, and then here you have, it almost looks like an ordered packing, but I don't know if there's any characteristic number of vertices. And then this area down here, which is I guess high P or low P bar, high Q, that's actually geometrically impossible. So that's an interesting kind of way to unpack some of this. No pun intended. It's just showing these different packing possibilities and what they look like. So that's, and then this is external deformation. So this is where you're forming this packing, and this is using cell alignment shape Q versus average shape index P bar. This is showing sort of under external deformation, the same result in figure 2. And the same thing for internal stress, which stretches this packing outward. and then you get the same result for Q versus P bar. So that's any other comments on that paper?
Just that I think I'm just going to quote it.
Yeah.
And let my friendly neighborhood Dr. Sharif will look at it if he wants to.
Yeah.
Or if he'll just accept the fact that other people are working on this problem.
Yeah.
Yeah. Why is it a hexagon? Why are you using a hexagon?
Yeah. Well, that's
I know everybody else does. Are you a PhD student or what?
Yeah.
I know.
Yeah. So I just want to talk about this paper briefly. This is a Nature Physics paper, collective curvature sensing and fluidity in three-dimensional multicellular systems. So this one is about collective cell migration, and I don't think this focuses as much on Drosophila, but let's see what they have to say. So collective cell migration is an essential process throughout the lives of multicellular organisms. For example, in embryonic development, wound healing, and tumor metastasis. Substrates or interfaces associated with these processes are typically curved. So there's this curvature aspect, that we talked about in Drosophila embryos. So if you look at the edge of the embryo there's a curvature, and that curvature has some implication for generating some of these forces, especially on the outside you have curvature, and even on the inside you have curvatures once the tissues are formed. So you get these, these are important kind of structures to hang. It's not, the embryo isn't a sphere. It's a sphere maybe sort of for a reason, but substrates or interfaces associated with these processes are typically curved, with radii of curvature comparable to many cell lengths. So there's this radii of curvature, which is like, how curved is it? Is it very tightly curved, or is it like, a oblong shape? And so using both artificial geometries and lung alveoli spheres derived from human-induced pluripotent stem cells, this sounds like some sort of organoid work, actually. Here we show that cells sense multicellular scale curvature, and that it plays a role in regulating collective cell migration. So this is where they're arguing that cells are sensing this curvature. They're sensing the curvature of this microenvironment, or the embryo, or whatever it is. And it's playing a role in regulating collective cell migration. So you can imagine that the cells, kind of stop at the boundary, or they kind of know that they don't flow outside of the boundary under normal conditions. They kind of find a boundary, and it's usually spherical, and there's a curvature to it, where otherwise we see in some of these hypothetical examples in the last paper where they used the square packing, and what we have actually in biology is a curvature. Furthermore, hexagonally shaped cells tend to aggregate in solid-like clusters surrounded by non-hexagonal cells that act as a background fluid. We propose that cells naturally form hexagonally organized clusters to minimize free energy.
Oh, yeah, this is perfect.
Yeah. And
just quote that.
Yeah, that part there.
That part there.
Yeah. And the size of these clusters is limited by the bending energy penalty. So there's this energetic aspect to it. So it's using free energy and minimizing that to form these spheres. And of course, we know from collective material, or the collective behavior of materials, that you have often have physical constraints to what the shapes that they form. So if you put, even if you put metal balls in an electric field, they can form different patterns. But they tend to do this not because they know what they're doing, but because that's the free energy path. The path of least energy, or the path of maybe balancing out magnetic forces or electric potential or something like that. So we observe that cluster size grows linearly as sphere radius increases, which further stabilizes the multicellular flow field and increases cell collectivity. As a result, increasing curvature tends to promote the fluidity in multicellular monolayer. Together, these findings highlight the potential for a fundamental role of curvature in regulating both spatial and temporal characteristics of multicellular systems. So this is where, we're trying to figure out how these get regulated. Okay, so this is, Susan says, order on curved surfaces. The curvature can greatly change ordering. A classic problem, packing spheres as tightly as possible, have a simple solution in flat space, leading to hexagonal packings in 2D and hexagonal close packed structures in 3D. On a curved surface, voids in the packing are required to accommodate the curvature. Our research studies and structure and location of these voids, now they are affected by non-uniform curvature in different sized particles. That's from Tufts, from the Tufts group, I guess. So, so let me see if we have any figures that we can go through on this. So this shows kind of the curvature. This is in A, it's a concavity, in B, it's a convexity, and then in C, you can see that this packing actually shows this. So this might be like a tumor in another tissue, and you have this concavity, the convexity, just showing the structure of this sphere, and then the sphere has a radius and an angle of curvature, which is here, and that angle of curvature is determined by the physics, but also it's, the cells can sense it, and they kind of know where these boundaries are once it's formed. And so you can have like say like a tumor or something that's spherical that sort of, the cells sort of form the sphere based on, minimizing their free energy. And then afterwards they can sense the boundaries of this. So they kind of have a guide aside from free energy as to where to go and where not to go. So this is the nucleus here. These are some nucleus stains where you can see the curvature of the cells, and you can see how tightly packed that is. These are MKCD cells or MDCK cells. So these are grown on different curvatures, and they're individual and collective cellular behaviors. So you have density versus velocity, and you can see this. This is where you have this value for K at different sizes, and you can see that they follow this trend here. And so this is the sphere here. This is the curvature angle. C sub W is, I don't know what that is exactly, but you can see that there's this trend here as well for this set of parameters. Yeah, great. And then this is another set of, so this is the multicellular flow field, and for MDCK cells, it's more dynamical with a larger curvature. So this is a divergence color map of this sphere and the divergence values along the surface of the sphere. This is an example of the sphere with its angle of curvature and divergence values across these different values of K, and then divergence versus the probability density function for each of these, and then this graph here. So this just basically shows divergence as the swelling and shrinking of cells, red representing sources and green representing sinks. So this is kind of showing like how they're moving around in this packing, and then so then this shows sort of the decrease of curvature over this. All right, so this is for the alveoli spheres, and so these are derived from human induced pluripotent stem cells. This confirms that collective cellular behavior is strongly affected by curvature. So you see here, this is a stain of this, where you have the extracellular matrix, you have these cells along the edge, and then the curvature decreases as it gets larger. And then you can see again some of these experiments where they show the divergence in these same structures. The divergence gets smaller as the packing gets bigger. The curl changes as well, it gets higher, and then the shear forces get lower as the packing increases. The shear forces are very high for these small packings. So the curvature is mediated by the size of the packing, and then these other parameters follow along. Okay, this is the reference for what Susan was talking about in that link. So I think that's all for today. Any other comments on that? I can send the papers. I know Susan probably found this to be very useful. Oh, go ahead. Muted. Oh, muted. Susan, you're muted.
We can't hear you.
Yeah, I got a phone call and turned it off. Anyway, yes, I could just hand the professor the references, the 2 or 3 references there, and say it's somebody else's research, and he probably will say fine, because he doesn't want me to redo somebody else's research.
Yeah.
Yeah, enough with the sidelines.
Yeah.
Yeah, especially the second one, and even Tufts University.
Yeah.
Stuff. It's good. I'll just quote what they've got, and yeah, they have an interesting oblong embryo that there's a simulation they're studying that looks like a fly embryo, like I said earlier, and it's there somewhere. And it's a misaligned packing in this oblong embryo, so it diverges from these hexagonal packings.
Right. Yeah. Yeah.
Anyways,
all right. Yeah. Well, glad you found that useful. Yeah. Sounds like fun.
Oh yeah. Saves me hours of work. I'll just throw it at him.
Yeah.
I can get on with the project.
Yeah.
And I'm going to try to find Morpho and use it. Anyway, meantime, this week I'm using MATLAB.
Yeah.
At least it has lots of support.
Right.
You can usually look up the error that it gives you, and it'll pop up with an answer to what you did wrong. Right. Yeah, it's very useful that way. It's got lots of support.
Yeah.
Unlike, say, console.
Yeah.
Anyway.
Yeah. So, Morgan, did you have something to say before you go?
Well, a really interesting paper, and I was checking out both the Morpho package, which be if I can be any use, let me know, Susan. And I think the most up-to-date reference that I think Dick put references in on kind of finite element methods in developmental biology, but in particular, I was interested in the 2017 reference that is trying to infer things about cellular forces from video stacks.
Oh, right.
And again, sort of relates to some other things that I know DevoWorm is interested in.
Yeah, you should come to the APS March meeting.
When is that? It must be this week.
No, it was 2 weeks ago.
I see, it's fast.
Yeah, you can attend virtually, and it's a lot less. Costs a lot less.
Yeah. I would be very upset if everybody goes back to non-virtual meetings.
Yeah.
Yeah.
Well, they had virtual meetings. I attend. Well, since I'm a student, I could attend for I think it was about $150 US to go to the virtual meeting. Or a student, I think maybe professors are $300 or some general public $300
Yeah.
US to attend the whole, the meeting for 5 days. I don't know. I think it's pretty good. Not
Yeah.
bang for your buck. It's
So is that a physics or physiology? What's the
Physics.
key?
It's physics, and you can do quantum physics if you want. I mean, they have lots of lectures on quantum physics, which I avoided.
Gotcha.
Yeah.
I mean, again, it's probably been about 30 years since I did my quantum.
Yeah.
No, and I don't want to relearn Feynman diagrams. Thank you. So I just this is soft matter, and it's under DSoft and what's the other one? I found the other one more useful. Anyway, there's 2 or 3 subheadings of that, and you can attend them. You just look at the lectures and see which ones you like.
That sounds good. And Bradley, you had a couple retweets of interesting things after we talked on Friday.
Yeah.
I think this weekend you might have had a couple interesting. I want to go through your feed.
Yeah.
Because I think after our talk there was a couple things that you posted that looked relevant too. I'm not remembering them right now.
Right.
But, I'm probably going to follow up with both of you about some references.
All right. Yeah, I'll see what it is. I think I don't really remember what it is right now, but I'll follow up on that.
Well, you're always posting interesting references.
Yeah.
Yeah. And true.
All right. Well, thanks for attending, and thank you to Sushmanth for his work on the web, and Susan for her conversation about this problem with packings and flows and things like that. It's a great topic. Thank you for Morgan for.
It's interesting.
Yeah.
Bradly, I want to mention one small thing.
Okay.
I will be not available for next 2 weeks for the meeting. I'm going on that village thing which I have mentioned in last meeting, that I'm going to a village to analyze their problems and all. I will not be able to attend the meeting, but I will give updates on Slack. And another thing you have mentioned that my proposal, the proposal is ambitious, and I just want to mention that thing proposal is ambitious, but it is like around 350 hours work.
Yeah.
I just want to. I'm just not doing this project as some kind of GSOC or what. I just want to continue this work as for my master's and my undergrad work also. So this is not an option for me. It's just I need to do it anyways. That's thing is there with me.
Oh yeah.
Whether it was selected for GSOC or not, I will do it as part of DevoLearn, post resolve instance segmentation on DevoLearn, and work on those DevoGraph also.
Yeah, it sounds great. Yeah, I mean, I just I always tell people
Okay.
well, yeah, but no, I think your proposal looks pretty good, and I definitely ready to submit.
And
so.
when Mayuk saw this comment, actually Mayuk has access to the doc. He told explain the situation like you're not doing this project. I mentioned him like I am not doing this project as some kind of GSOC thing. I'm doing this because like I want to extend this as my master's degree, studying this domain only, neuroscience and machine learning.
Okay, well, yeah, that's great. Yeah, I can help out, any way that you need for that too. So that'd be good.
Okay. Thanks. That's one. I will not be available for next 2 meetings. So
okay, yeah.
If anything is there, I will mention in Slack.
Yeah, and I actually we won't be having a meeting next week. I have a scheduling conflict, so yeah.
Okay. Thanks.
Doing great.
All right. Yeah. All right. Take care.
Yeah.
Thank you.
Bye.
Bye.
Thank you.